Welcome to the Neuroveda podcast for complex health, where we feature expert guests who dive into ancient and cutting edge understandings of health and disease. These conversations are about food, lifestyle, and medicine just as much as about nature, culture, and politics. I'm Gillian Ehrlich, family nurse practitioner, certified in Ayurveda and functional medicine, coming to you from our clinic, Neuroveda Health, in Seattle, Washington. And I'm pretty thrilled about our guest today. I know I say that every time, but it's like I am really thrilled about every guest. And our topic today is oxidative stress, which is a big topic, a complex topic. And we're going to dive in with Dr. Jessy Dhanjal. He's a licensed naturopathic doctor in California. He earned his doctorate in naturopathic medicine from Bastyr University in San Diego and holds a Bachelor of Science in Biological Sciences from the University of California, Davis. During his time at UC Davis, he participated in the Emergency Medicine Research Associate Program at UC Davis Medical Center, gaining early clinical experience in the emergency department. Dr. Dhanjal has pursued advanced training in functional medicine through the Institute for Functional Medicine. His clinical interests include primary care with an emphasis on cardiometabolic health, autoimmune conditions, gastrointestinal health, and detoxification strategies for environmental toxin exposure. Currently, Dr. Dhanjal serves as a clinical lab educator at Vibrant Wellness, where he supports healthcare providers in understanding and applying advanced diagnostic testing to enhance patient outcomes. He believes deeply in the body's innate ability to heal and is committed to helping patients restore and maintain optimal health. You can find Vibrant at vibrant-wellness.com. Dr. Dhanjal, welcome. We're so happy to have you.
Thank you. Really appreciate it. Thanks for having me.
Yes. All right. So let's dive in. What is oxidative stress and why would we care about this for human health?
That's the $1 million question, right? And we hear this term a lot, right? But before we begin, I definitely want to talk about what Vibrant Wellness is.
Oh, yeah. Please
What
do. Please do.
But basically, it's a lab. It's not your average lab. At its core, it's a technology-driven company. They use cutting-edge, high-quality technology to deliver reproducible, informative lab results. And really, it specializes in advanced functional testing that goes upstream, right? Looking at genetic predispositions, real-time biochemical dysfunction, toxic burden, nutrient depletion, gut microbiome balance, Lyme infections, and more. So it's a CLIA certified and CAP certified lab, which means basically the standards are met with to the highest level for clinical testing. But what makes Vibrant really unique is that we're research first. So Vibrant's heavily in peer-reviewed research, collaborating with John Hopkins, Mayo Clinic, Cleveland Clinic to validate panels and technologies. So ultimately, it's to empower clinicians and patients also with actionable personalized health insights through lab testing. Oh, oxidative stress is, we hear that all the time in the news or conferences, but really, what is it, right? The simplest way I can define it is it's an imbalance between free radicals, your unstable molecules that we hear, of oxygen species, even reactive nitrogen species, and the antioxidant defenses that are meant to keep them in check. Often I hear, what is a free radical, right? I get that question a lot, but think of that as a molecule with a missing puzzle piece. It's an unbalanced and will do whatever it takes to find the missing electron to balance itself out, right? Two of the uh, most famous free radicals you probably hear about is superoxide or hydroxyl radical, right? So if we go back to maybe chemistry, these stable molecules have a full outer shell electron, right? They're cool, calm, collected. Once that stability is lost, that molecule becomes highly reactive, and all it wants to do is just find something to stabilize. And I always like to explain to patients and providers that free radicals aren't inherently bad. They're actually normal, right? A byproduct of metabolism. And the body uses them for many different functions, for example, immune defense, right? But problems arise when they start to accumulate faster than the body can neutralize them, right? When that happens, free radicals can start attacking key cellular components, lipids, proteins, DNA, even mitochondria, right? And that's where oxidative stress kicks in. So this matters because oxidative stress is a root contributor to almost every chronic disease you can name: cardiovascular disease, neurodegeneration, autoimmune issues, right? Diabetes, I guess even aging, the process itself. So the best way I can describe it, it's kind of like a silent inflammation, gradually wearing down your body's systems over time. Now, you may not feel oxidative stress directly, but patients may have signs of fatigue, brain fog, joint stiffness. But underneath, quietly oxidative stress may be driving dysfunction. So, that's why we offer in our treatment options antioxidants. They donate those electrons to free radicals to basically stabilize them.
It's
Yeah.
so hard not to make political jokes about free radicals right now with our government is standing, but I'm going to refrain because I think what I'm hearing, which I think is so critical to human health, to the individual body, as well as our collective bodies, is that our metabolism, we do change over time, right? I often use with patients that analogy that in 7 years, I don't know if this is actually true, but by 7 years, every cell in our body is different than it was 7 years ago. We have patterns of cellular tissue that are the same, but when we think of our identity, we are not just like a person who grows, reaches adulthood, and then never change. Our skin cells, I know our GI tract turns over every 3 weeks-ish. Our neurons can last the longest, but it is like these new cells get taught by the cells that are present how to function within that milieu and in that environment. And so these patterns really can persist. And to make any change, you essentially, like even in remodeling a house, you can't put a new roof on before taking off the old roof. And so you're constantly needing to break things down. And these free radicals can be really helpful either as byproducts or as triggers or as results or as things that need to be eliminated. And so it's part of that process of regeneration that we are doing lifelong, from like before our first breath until we like decompose after our last breath. And so this is a great primer and reminder on like the elemental importance. Like we cannot live without free radicals. And like no matter of perfect diet, perfect sleep, perfect exercise is going to prevent you from having these natural and appropriate free radicals in your system.
You got it. That's exactly it.
All right.
Right.
Okay, perfect. And so we need these free radicals. They're appropriate. They do some really important things. And then sometimes they like just go sideways.
Yeah, exactly. And you have to kind of think about what causes this in the first place, right? Like you just mentioned, right? They're naturally produced, and it's part of our normal cell functions, right? Most of it's happening during our aerobic respiration, especially in the mitochondria, which are cells, how they generate energy. Part of our immune system as well, called macrophages. They create these reactive oxygen species to help defend the body. And that's when macrophages engulf, these harmful invaders and they release these ROS to kill these pathogens. So, but, ROSs aren't just made during infections. They're also produced from inflammation as well, even if no pathogens are around, right? So that means during chronic inflammation, macrophages can keep up, keep making ROS constantly. That's where it can lead to oxidative stress and damage to tissues over time. And then I always like to mention, on top of the internal sources, we're also exposed to from the outside of the body. Think things like heavy metals, environmental toxins, right? Mold toxins, mycotoxins, and I guess even certain foods, right, can increase reactive oxygen species, especially, metals like cadmium or nickel, right? We offer the heavy metal test, which tests for what patients have in their body, right? What they've accumulated metal wise. And those metals can cause some disruption. I mean, create superoxide and hydroxyl radicals. And basically, right, with oxidative stress, I'm probably going to be hammering this throughout this talk, right? When ROS levels get too high, right? As you can see, it come from both inside and outside the body, right? So that's where we as integrative functional health providers, we have to take a detailed medical and lifestyle history to, hey, spot that possible exposure, right? So, have they been on medications? Are they near environmental pollutants, right? Living wise or career wise, right? Alcohol, UV light, they've been smoking cigarettes. It's a major cause of lung cancer, especially with combined with genetic factors. And then I guess also inflammation, right, is another big contributor to oxidative stress, which can be triggered by external factors. Often also I mentioned, metabolic issues like high blood sugar, that can also generate oxidative stress. So that stress, right, like I mentioned before, will harm lipids, DNA, RNA, and proteins. That's an exciting thing that Vibrant offers is an advanced test that can measure these oxidative damage across all these different molecules, right? Before, we mainly relied on the 8-OHDG, right, 8-hydroxy deoxyguanosine, just to detect DNA damage. And industries come up so fast now, so there's many markers that can assess damage to these proteins, lipids, DNA, and gives you a fuller picture of oxidative stress and how to affect the body.
Amazing. Amazing. And so, and one way to think about the impact of these free radicals is kind of like aging or inflammaging, sometimes we say. And they really, if we have an imbalance in the production and the like the soothing or the quieting or the calming of these free radicals, if we have an imbalance, then we're just going to have increased inflammation and we're going to age faster. Is that right? Sometimes I think about oxidative stress. I describe it to patients as like we're rusting from the inside.
Oh yeah, that's a great analogy to use actually. And that's why these tests, right? It kind of gives you, when you look at the genetic predisposition, and if you have some abnormality, right, it's kind of like smoke coming out. There's no true fire yet, but we come in as firefighters to kind of make sure how we can help the patient, right, through nutrient strategies and kind of provide that kind of genetic bypass.
And so, how does oxidative stress, how is it connected to chronic disease?
Yep, definitely. So there is a connection, maybe an indirect way, but there's a lot of research showing how it's involved in many different health conditions. I think two of the big ones I always like to talk about, especially it comes up in integrated medicine or cardiovascular, right? Autoimmune disorders, especially when you think of atherosclerosis, right? Oxidative stress is a major factor, right? It causes problems in the lining of blood vessels, endothelial dysfunction, right? That dysfunction starts and pushes the disease forward, leading to things like the growth of endothelial or smooth muscle cells and narrowing of blood vessels, right? So those are key features of atherosclerosis. And, with our cardiac health panel, we actually test the myeloperoxidase enzyme, right? That's produced by our immune cells to kill pathogens by generating reactive oxygen species, right? But excessive MPO, that can lead to oxidative stress, damaging these tissues, contributing to atherosclerosis and inflammation. So oftentimes when we see markers elevated, you have to balance that, right, to prevent tissue damage while maintaining also that immune defense. And then the other option, autoimmune disease, right? So it works a bit differently. Oxidative stress damages proteins, fats, DNA. It creates these altered molecules called new epitopes. Basically, the immune system mistakes these as foreign invaders, right? And then they produce these autoantibodies against them. And that fuels ongoing inflammation and worsens autoimmune activity. So, that process basically shows oxidative stress is central to many chronic diseases. But, there's many other diseases out like aging you mentioned, right? It's closely related to oxidative damage, right? Problems like with muscle loss, heart disease, right, which I mentioned, lung cancer, especially COPD. But there's macular degeneration, metabolic disorders, diabetes, fatty liver disease. I think there's even growing evidence now to mental health conditions like depression, bipolar disorder, schizophrenia. So it pretty much touches every aspect of the body. And we as healthcare providers, we have to really understand oxidative stress, right? Especially when working with these conditions. And that's where this test can help us measure these oxidative damage in patients, right? Kind of guide a more focused and tailored treatment because this topic can get pretty complex, right? Especially inflammatory cells are central to many chronic diseases. And when these cells are activated, they release lots of reactive oxygen species. And that leads to more oxidative stress, more tissue damage, right? And I think what gets trickier is when high ROS levels can activate these inflammatory signals, right? They kind of feed off each other. It's like a classic chicken or egg question, right? It's hard to say which come first, right? But with good testing and definitely careful history taking, we can start to see figured out in the patients, right? So that's an important paradox, right? Just like inflammation, there's a certain amount of RS that's needed, which you mentioned, right, is actually good, but it can destroy microbes, help remove damaged cells, right, apoptosis. But the problem is when it gets too high and it's not well controlled, and that can drive chronic disease development and progression.
And so, talk to us a little bit about the role of genetics. Like, how do genetics change or impact oxidative stress?
Yeah, so absolutely. Genetics can significantly impact how well our bodies handle oxidative stress. Now, that's different within everybody, right? And with this oxidative stress panel, right, we can actually see what's going on. And, while the lifestyle choices like diet, sleep, exercise can definitely influence that balance, the genetic makeup of an individual plays a foundational role in determining how efficiently those antioxidant pathways work, right? So in clinical practice, we often see this play out in kind of real ways, right? For example, you might see a person who's doing everything right. They eat clean, they don't smoke or drink, they exercise regularly, right? Good sleep, not too much stress, and they're still showing signs of elevated oxidative stress, right? So in those cases, we often find unfavorable genetic variants that impair these antioxidants defense mechanisms. On the flip side, there are people whose lifestyles aren't great, right? They might be eating poorly, stressed, right, through work, family, right, skipping workouts, but their oxidative stress markers come back stable. So
Mhm.
that could suggest strong genetic function in those detox and antioxidant pathways. It kind of gives them that built-in buffer or protection. But when we look at genetic testing, you have to think about nutrigenetics, right? So nutrigenetics focuses on how your genes influence your response to nutrients, right? So everything
Mhm.
how you absorb, metabolize, and utilize these key vitamins and minerals to help to effectively neutralize these free radicals. So instead of a one size fits all approach to health, you can tailor interventions based on someone's unique biochemical profile, through testing.
So
So
cool.
we mainly look at variations that influence these enzymes, right? So, for example, looking at SNPs, single nucleotide polymorphism. So, these are common variations in the DNA, right? Small single letter changes in the genetic code. While some SNPs have little or no effect, the others, other SNPs can particularly affect how an enzyme works, right? So, to put it in perspective, most humans have about 4 to 5 million SNPs, right? But only a small subset are relevant to specific biological pathways. And with the oxidative stress panel, we look at almost 32 different SNPs that either directly are involved in neutralizing these free radicals to help recycle endogenous antioxidants. So think of glutathione, superoxide dismutase, right? Those are critical to oxidative balance, right? So we often supplement those to patients, right? Glutathione, we hear all the time, right? Pretty much ever, it's a master antioxidant. But if you think about it, right, it's made up of 3 amino acids: glutamine, cysteine, glycine, and super critical in protecting cells from oxidative stress, detoxification, immune function, protein structure. And even on the report, it talks about, basically glutathione exists in two forms. And there's a diagram on the report, but reduced form of glutathione, we often reference it as GSH. So that's an active antioxidant form. And then the oxidized form is GSSG, which forms when GSH neutralizes these free radicals, right? So almost in all healthy cells, 90% of glutathione is in the reduced form, so GSH. And the body uses enzymes like glutathione peroxidase, which uses GSH to reduce these harmful compounds like hydrogen peroxide, right? And then glutathione reductase, which regenerates GSH from GSSG. So our body's really neat in that aspect in making sure glutathione is recycled properly. And, that's where we think about conditions like such as Alzheimer's, right? Which I'm sure you're aware of, cancer, right? Cardiovascular disease. And then these GSH levels tend to decline, right? And then that's where we as providers have to think about supporting these GSH levels, uh, to be an important strategy for reducing this disease risk, especially through genetic SNPs. And then another key enzyme I just mentioned earlier was superoxide dismutase. So these are a family of antioxidant enzymes. So they're pretty much found in all aerobic organisms, but its main role is to protect cells from oxidative damage, right? Converting these superoxide radicals into hydrogen peroxide and oxygen. So these superoxide radicals are byproducts, like we talked about, right? Cellular metabolism, right? Mitochondrial respiration. So, for example, on the test, we look at SOD1, SOD2, SOD3 enzymes, right? So each of these, I'll just quickly talk about. SOD1 is primarily found in the cytoplasm and uses copper and zinc at its active site. It's a general antioxidant. It's involved in carbohydrates, protein, fat metabolism. Even SOD1 mutations are known to be linked to ALS, actually. And then SOD2 is located in the mitochondria. That uses actually manganese as a cofactor. So now you can start to see how micronutrients are beneficial, right? So making sure those are at optimum levels to even help with these enzymes, right? And as you know, mitochondria is a major source of reactive oxygen species due to the electron transport chain, right? So when we see enzyme SNPs or deficiency, right, then you have to start thinking about mitochondrial dysfunction. Is that increasing oxidative stress, right? Potential damage to mitochondrial DNA. So that's linked to aging, neurodegeneration. So, because of its central role in mitochondrial health, I think it's even being studied now with particular conditions like ischemic brain injury, actually. And then the SOD3 is found in the extracellular matrix. So that's pretty much in blood, lungs, brain, and vascular tissues. That uses copper and zinc like SOD1. But think of this as protects, the organs, heart, right, brain, blood vessels. So it's more of immune response regulation. You can find these supplements out there, right? SOD2 or SOD3, good combination formulas. But back to the SNP, right? In the report, we look at a specific RSID. So that's a unique reference number for these gene genetic SNPs. So for example, on the test, you might see like RS for 880. That's basically the SOD gene, which plays a role, right, in converting these superoxide radicals into less harmful molecules. So with genetics, right, when we look at these SNPs, right, you have to look at the patient's genotype in three ways, right? So the test actually will lay this out for you. So there's homozygous wild type, there's heterozygous, and homozygous variant, right? So homozygous wild type, that's the most common form in the population. That's considered the reference or typical version. Doesn't mean it's always the best, but it usually indicates normal function. Heterozygous, that's usually with the plus or minus. That means one copy of the gene has a reference allele and the other has a variation. So depending on that specific gene and the nature of that SNP, that could slightly reduce that enzyme function, right? So you may not be conjugating glutathione or not producing enough glutathione as optimum, right? You're in the middle there. And then homozygous variant is when both copies of the gene carry the variation. So you will see that greatest functional impact, right? Reduced enzyme efficiency and therefore higher need for targeted support. So that's why we have to understand these distinctions, not just for providers, but educating patients, because we're not just talking about genetic mutations here, right? Mutations you think are rare, right? Associated with specific disease. What we are looking at is common genetic variants that influence the function, right? Not disease in a traditional sense. And I think the last part I'll say is I always like to kind of use analogies. Think of these as biochemical pathways, like highways, right? Some people have clear, open routes where things are moving smoothly, and others might have partial blockages, right? Think road work or reduced lanes. And then some might have severe congestion, right? So our goal is with testing is to identify where those bottlenecks are at, right? And figure out how to bypass that, which I mentioned earlier, right? So if somebody has an impairment in glutathione recycling, we might recommend specific antioxidant nutrients or cofactors to compensate for that weakness, right? Create that genetic bypass. So we're looking at the body's blueprint to personalize interventions and outcomes. So yeah, the approach is not just by diagnosing disease; it's about optimizing health at the cellular level. And that's where I think we try to empower people with information to make targeted changes to improve resilience, right? Prevent dysfunction before it starts. So to answer your question, absolutely genetics.
And so genetics, it's interesting because a lot of times when we think about genetics, I think a lot of us have been brought up with the idea that it's like one gene is a problem. And it is true that there are genes that we think of as pathogenic. So sometimes that might be like the gene for sickle cell anemia or the gene for Huntington's chorea, or there, but the majority and the truth is that so many genes play into our health status, and that the body, to be so resilient through, hundreds of thousands of years of evolution, really has so many redundancies built into the system. And so when we talk about SNPs, single nucleotide polymorphisms, we're really talking about those highways. It's kind of like you can still get to your destination, but you may have to take a different route. And if you have to take a different route enough times, it really can slow down your productivity at work. If you're only, if your 8-hour workday is shortened to a 4-hour workday. And so it's really interesting to talk about these particular group of SNPs. And so when you do, there are two parts to this oxidative stress panel that we're talking about today. So the first part is the genetics and the SNPs. And then the second part is the actual metabolites to look at the function, because the truth is that once you know your SNPs are not going to change.
Going with
You have them for life. And so once you know them, you know them. And once you know them, you can really address them and how you like live your life and through your diet and your lifestyle. And I look at them a lot with patients to know, how hard do we need to work?
yeah.
Because the idea is like, it is not, when you think about how many personal care things you can do for yourself in a day. I mean, we have a list. I mean, we have a joke in Ayurveda. I mean, it's not totally a joke, but there's our teacher, Dr. Vasant Lad, has put together a daily routine that includes like 17 things for the morning. By the time you leave the house, there's 17 things that you could do. And some of them are like emptying the bowel and bladder, or, oral hygiene, which can include scraping the tongue and brushing the teeth and oil pulling and rubbing trifla on your gums. There's 4 things just for oral hygiene. But all of that to say that there are so many things that we can do. You can only do so many things in a life, and our lives in different moments are going to demand different activities of us. So you can't say to a person caring for a newborn, make sure you get 8 hours of uninterrupted sleep at night, because guess what? It is going to be interrupted. And so we have to make these choices, and these SNPs can really help give us perspective on how hard do we have to go for anti-inflammatory diet, lifestyle, supplement, medication support, all those components.
Yeah, definitely. And that's the cool thing about our body, right? How does it normally defend itself, right? Our bodies have a really sophisticated built-in defense system, right, to handle oxidative stress. One thing we teach is a molecule called NRF2, right? Maybe you've heard of that, but basically it's a master switch for antioxidant protection, right? So under normal conditions, NRF2, which stands for nuclear factor, I think erythroid 2, related factor 2. It's a mouthful, but it just kind of hangs out in the cytoplasm in an active state. But when the body senses oxidative stress, right, it's so wise that whether it's from internal sources, from like hydrogen peroxide or reactive molecules, it's like an alarm that goes off, right? And when that gets activated, it moves into the nucleus of the cell. And that's where it binds to the DNA, right? We call it the antioxidant response element, short for AR. That's basically the command center to what signals the cell to start producing a whole range of these antioxidant detoxification genes. And that's where these genes can help neutralize these reactive oxygen species, right? Restore balance, protect your cells from damage, right? So in a way, think of that as your body's internal antioxidant thermostat, right? So, all those different lifestyle aspects you suggested, right? But even diet, right? We focus highly on phytonutrients, right, from plants, right? These natural compounds that contribute to their color, their flavor, right? They're resistant to pests. Those can also activate the NRF2 pathway. And think of sulforaphane, right, from broccoli or cruciferous vegetables, curcumin from turmeric, green tea, right? Beets, right? Betanin, the natural pigment that gives that red deep color, resveratrol, right, from grapes, quercetin. So you can get that through foods as well and supplements. So when we talk about defenses, right, we're really referring to two categories. So enzymatic and non-enzymatic antioxidants, right? These enzymatic antioxidants is what I mentioned earlier, right? They're made by our body to neutralize these free radicals. So think of a superoxide dismutase, right? Catalase, right? Catalase breaks down hydrogen peroxide into water and oxygen. The end game of the body, what it's trying to do with these free radicals is to just make it lipid stable and water stable. So it's just balanced, right? A glutathione peroxidase, right? Glutathione reductase. So both use glutathione to detoxify harmful compounds. So think of the antioxidant army as your first responders, right? They're fast, efficient, working behind the scenes. And then importantly, we also have non-enzymatic antioxidants, right? So in addendum to lifestyle, right, you can also supplement accordingly, right? Glutathione, like coenzyme Q10, right, especially for cardiovascular issues, alpha lipoic acid, right, melatonin. Yep, the sleep hormone. It is a very powerful antioxidant, right? So when you combine all that, that's how you protect cells from damage, right? Kind of a with our own, innate system, right? Plus external what we can do. It's like a well-coordinated orchestra with different sections playing different instruments, but it's working towards the same goal, right? Keeping you resilient and balanced. That's how at least I like to frame it.
Amazing, amazing. And so the SNPs that we're looking at are really in the category of the superoxide dismutase, so SOD1, 2, and 3, and then the glutathione pathway. So there's GSS, glutathione synthetase, GST, glutathione S transferase, GPX, glutathione peroxidase, and GR, glutathione reductase. So we've got 4 of the glutathiones, 3 of the SODs, and then we've got a CAT or catalase. So anything else we should talk kind of about these
Yeah.
SNPs before we go on to the
Very
metabolite?
complex.
Okay.
Yep. And that's the best part. It's always you got to start with genetics 101, especially when you're looking at these type of genetics. That's where this oxidative stress panel that Vibrant offers is incredibly comprehensive, right? It's not just looking at oxidative stress, but you have to start thinking why, where, and what, right? What to do about it. So with Vibrance technology, we use kind of two advanced methodologies, which I'm sure you've heard right in past podcasts, right? So we look at genetic SNPs using PCR, right? Real-time PCR, right? Which is pretty much a gold standard that allows for 100% accuracy and reproducibility. And then to measure the oxidative damage markers like dityrosine or advanced glycation end products, that's where they use liquid chromatography with tandem mass spectrometry. If for folks who don't know, right, PCR is basically it's a molecular biology technique used to measure specific genetic sequences, right? In this case, SNPs. So the way it works is, you take some type of DNA sample. It could be saliva, right? You extract it, and then you use the primers to amplify it and really get to look at the regions of interest. And when you amplify, right, millions of copies, that's when it gets easy to detect, right? And these machines are so smart these days, right? Using fluorescent markers to track how the DNA is amplified. And then in mass spectrometry, we back to organic chemistry, right? And that's where they use the looking at the sort of breaking down the compound, right? Looking at how much it weighs and identifying them, right? With extreme precision. So definitely it's technology matters, right? And that's where it comes down to looking at these four kind of major sections of how the stress panel is divided, right? So I'm probably going to hammer again on the genetic SNPs because it's that important, right? So on the first part of the test, there's a flow chart of your antioxidant pathway. It summarizes kind of the main SNPs as you just suggested, right, with the single nucleotide polymorphism, so you can see where things are kind of bottlenecked, right, with the highway analogy. And then the second part of the genetics on the test is all the list of variants showing how enzymes are functioning, kind of looking at the traffic pattern on the highway. Are the antioxidant roads wide open? Are they flowing smooth? Are there any blockages, right? Congestion and backup. And that's where we have to put our thinking hat on as providers to, future think what we can do to help with at least treatments, right?
Mm.
And that's where you'll see that RSID number, right, which I mentioned. And then the gene name, right, the genotypes. So you get all that information, right? And then you get the risk association. So it tells you how that variant may impact activity, right? So homozygous variant, right, means higher risk for reduced enzyme activity. And then there's a middle heterozygous and then homozygous wild type, right? So we're essentially mapping out whether your antioxidant defense system is functioning like in a major 6-lane freeway or something. To the enzymes that you mentioned, right? One of the most important ones is actually glutathione synthetase, right? That enzyme helps your body make a glutathione. It's considered a master antioxidant. So if a patient has a homozygous variant, right, in that GSS gene, that means that really severely reduces the ability to produce glutathione. So even if you give, for example, glutathione precursors like NAC, right, N-acetylcysteine, it might not help because the enzyme that assembles those pieces is impaired. So that's where we as providers have to start thinking, okay, let's give them potentially bioavailable glutathione directly, right? Liposomal or IV if you practice that way, and assess the toxic load since a higher toxin burden increases glutathione demand, right? And then the other key enzymes you mentioned, right? Glutathione peroxidase, its main job is to break down hydrogen peroxide into water, right? If that's impaired, due to a SNP, that hydrogen peroxide can build up, creating more oxidative damage, right? And that's where we look at the next part of the test, what actual biomarkers are being damaged. And then catalase is another enzyme that also works with glutathione peroxidase to clear hydrogen peroxide. So you can get supplements with catalase out there, right? Obviously glutathione. So you have to start thinking in that manner, right? It's like when the detox highway is shut down, right? The oxidative stress is building up. How can you help, right? That's kind of a highway traffic cop. You could think of it as you're being at like a GPS app, right? You're looking at the patient antioxidant
Yeah.
ways, you're looking for roadblocks, kind of figuring out where to go. So for example, like GPX1, maybe you'll use selenium, vitamin C, right? Vitamin D, right? Think of those as your cops or construction crew, right? And catalase is actually vitamin E, right? Selenium, CoQ10, right? So those will help support mitochondrial function. And then there's the main part of the test, right? The third and fourth part. So we're looking at the actual damage from those SNPs, right? And why there's a buildup of those oxidative markers, right? So we're looking at now the footprints that's left behind, right? The biological damage, the DNA damage, right? The lipid peroxidation, protein oxidation, nitrative, right? Glycation stress markers. So Vibrant measures pretty much 16 specific biomarkers. They're grouped into 5 categories nicely on the report. And that gives you an insight, right, into different systems. So for example, you should be thinking, right, lipid peroxidation, right? Can that be pointing towards cardiovascular issues? Is DNA or RNA oxidation with those mutations, is it associated with aging or cancer risk, right? Like breast cancer, protein damage, right? Is that impairing metabolic or other structural functions? And then there's also AGEs, we call them advanced glycation end products. You can get that through the diet. Those are tied to blood sugar and even diabetes risk, right? So it gets really specific. And the most important thing to think about is, we're looking at more of a theoretical concern based on the genetics, right? And then maybe even an active problem that's causing the tissues right now, right? So basically, I think if we look back a little bit, I don't want to hammer on oxidative stress as a bad thing, right? It's not always bad. It can
Right.
help actually, right? That's the most important thing. I don't know if you've heard of a terminology called hormesis, right? So
Mhm.
basically, low levels of stress kind of trigger a protective mechanism that builds resilience, right? So think of when we see these kind of markers, right? Like I mentioned, a smoke alarm test, right? It keeps your system alert and response, right? But when it gets chronic or excessive, that's where it becomes harmful, right? And activating, like we talked about, inflammatory pathways, causing cell damage, mitochondrial, right? Even apoptosis, leading to issues, right? Health issues specifically. So that's why we just don't, as providers, don't hand out antioxidants to everybody. You have to use the data to personalize these interventions, right? The genetic makeup, oxidative stress burden, right? Lifestyle, toxin, right? Nutrient status. So we test before we treat. That's what I always like to say.
Absolutely, absolutely. And these biomarkers, the metabolites, are really broken, as you were saying, into the 4 categories. So let's talk about each category individually. So they break down into lipid peroxidation, DNA damage, RNA damage, and advanced glycation end products. So let's start with so out of the lipid peroxidation, there's 5 markers there. Talk to us a little bit specifically about
Yeah,
the oxidative stress that results in lipid peroxidation and what those markers are and kind of what we can do about the lipids if they're
definitely.
oxidized.
Definitely. Yeah. So there's a robust amount, right? There's 16 biomarkers. And, if you're always keen to learn on more, definitely reach out to us. We can go with it individually. But yeah, let's start with I think I mentioned OHDG first, right? That's kind of the most well-known established marker, right, for oxidative damage to DNA. We actually have that included in our hormonal zoomer, right? So it forms when reactive oxygen species basically these unstable molecules could be through stress, right? Toxins, as we mentioned, they damage guanine base in your DNA. Actually, guanine is the most susceptible to oxidative stress, right? So when that happens, your body is trying to repair the damage, right? And excretes 8-OHDG in the urine. So, elevated levels, if we see that in the urine, tells us that your DNA is under oxidative stress, right? And your body is actively trying to repair it. So it's not diagnostic of anything, but these are early warning signals, right, for most of these biomarkers. So it's useful for monitoring response to interventions, right? Detox, antioxidant, right? Lifestyle changes. So that's important. Then there's, you mentioned fat, right? Lipid peroxidation, right? One of the most clinically useful markers in this category is 8-isoprostaglandin F2 alpha, right? It's another mouthful, but in the cardiovascular health, right, it's an important marker, right? These markers form when fats in your cell membranes are damaged by oxidative stress, right? Particularly a type of fat called, for example, arachidonic acid, right? Arachidonic acid. These are unstable, unlike these free radicals that are unstable, right? These compounds, when we measure them, right, you can really see how much oxidative stress is damaging the lipids once we see this in the urine, right? It's especially useful in cardiovascular health, even inflammation, right? So you have to start thinking treatment wise how we can support cellular, right? Lining, right? For example, like phosphatidylcholine, right? So even with protein oxidation, there's another key marker, dityrosine, right? This forms when oxidative stress causes two tyrosine residues and proteins to bind together, right? Results in sticky, kind of tangled protein structures. So that damage is actually important for the brain, right? Linked to neurodegenerative diseases like Alzheimer's. So if we see elevated markers like dityrosine, right, it tells us that, hey, oxidative stress might be impacting neurological function, right? So that maybe opens up the door for interventions for protecting the brain, right? And the brain is a good source of fat, right? So that's when you combine these markers together, right? You can really come up with a good strategy and help the patient at least preventively, right? Or even during, right, if they have some type of chronic issue, right? So that's where these actual biological markers are super important to actually see what the body is producing. Right. And there's a couple other categories I'll go over quickly, right? The nitrate of stress. So these reactive nitrogen species are really unstable, highly reactive molecules, right? They usually form from nitric oxide and other nitrogen compounds. Unlike reactive oxygen species, right, these RNS contribute to oxidative stress where they build up faster, right? And the body can neutralize them with obviously antioxidants, but they can still build up with certain genetic SNPs, right? And when that happens, right, it'll damage your DNA, right? Fats, proteins, and these common RNS free radicals like nitric oxide and nitrogen dioxide, and even non radical forms like peroxynitrite, right, which forms when nitric oxide actually combines with superoxides, right? So RNS can still be helpful, right? They support normal blood flow, immune defense, communication between cells. But when levels get high, they can trigger harmful changes inside the body, especially type of damage we call nitrative stress. So that's importantly, definitely in chronic inflammation or even infections like H. pylori, right, where RNS can lead to long-term DNA damage and increase the risk of diseases such as cancer, right? One of the markers that we test is 8-nitroguanine, right, a compound that forms when RNS and DNA, right? So when these elevated levels of this markers, you have to really look at what's causing this at a deeper cellular harm is what I'm trying to say. And then the last category is advanced glycation end products, right? So this one I really like to emphasize on because it comes through actually diet, right? some of it, but it's also internalized as well. So there's two key markers we look at: CML, which is carboxymethyl lysine, and CEL, which is carboxyethyl lysine. So these compounds are when sugars react with proteins and lipids and DNA, right? Kind of a non-enzymatic process we call Maillard reaction, right? It's basically when sugars react with proteins and during cooking or high temperatures, kind of that browning, right? That flavor development. When these markers are really studied, and like I said, they're mainly formed from oxidized sugars and lipid peroxidation as well, which you mentioned earlier, right? So CEL is derived from methyl glyoxyl. It's a reactive byproduct of glucose and fructose metabolism, right? Both come from our diet, and then it's produced under the body through oxidative stress. And these advanced glycation end products, they interact with our receptor called in our body RAGE, right? That stands for receptor for advanced glycation end products. And that receptor RAGE is expressed in many different cell types: macrophages, endothelial cells, right? Epithelial cells. And they activate inflammatory pathways, right? NF-kappa B. So triggering oxidative stress through NADPH oxidase, for example. So what I'm trying to get at is this leads to production of those free radicals: superoxide, right? Peroxynitrite, a reactive nitrogen species. So there's a vicious cycle of inflammation, oxidative damage. So in clinical terms, right, you may see a patient history, you need to start thinking about atherosclerosis, right? This can stiffen the blood vessels by cross-linking with collagen, right? Hypertension, right? Altering LDL cholesterol to make it more atherogenic, right? Intervening with cellular signaling. And even to an extent, the gut barrier and microbiome, right? We see in conditions like an IBD. So diet is another major source of this as well, right? So when we cook meat or dry heat, right, baked goods or grilled or even snacks, right, those are high in CML and CEL. So that's why, cooking as through steaming and boiling produces less of these advanced glycation end products. So really, with all these different categories, right, as you can tell, it's quite a bit. And these markers give us a detailed picture of what's going on in the body, right? DNA damage, fat oxidation, protein dysfunction, right? Inflammation driven damage, sugar related stress, right? So we can track these over time to see how well the body is responding, right? And then the final component on the report is the oxidative damage score. So this is our proprietary algorithm. It evaluates all the 16 biomarkers on the panel, right? And then the reference ranges were established using a cohort of 1,000 healthy individuals. And that score is visually represented in the patient's chronological age. It's not a biological age, but the goal is to maintain a score at least 5 points below in one's actual age, right? So when we look at that graph, placing the individual in the green or optimal zone, that corresponds to the 50th percentile of oxidative stress. And then scores in yellow zone indicate more oxidative stress, up to 90th percentile. So while those in red zone reflect a higher oxidative 90th percentile. So it's a good, nice visual representation actually to show patient up front, really, because this is all complex biochemical data, right? And it's a nice, easy understand format. So you can really monitor the progress over time, right? And for many patients, when they see their scores improve, right, that's strong motivation to stay consistent with treatments and lifestyle changes, right? Initially, when they may see this type of marker score, they might get shocked, right? But that could also trigger motivation, right? And kind of adjusting and being really inclined to finish the treatment.
Great. And how quickly do these markers change? So, like when we're talking about instigating an intervention or initiating an intervention, how soon, like, could these markers change? Like, if you were in a car accident on the way to your test, on the way to your, to check this, could your stress levels be so high or like, does it happen that quickly? And how soon after an intervention is it recommended to recheck to see kind of a gestalt change?
So there's not really a good answer for that. It just depends, right?
Mhm.
Now, everybody's genetics are different. Everybody's toxin burden is different, right? So you have to look at other parts of the body, right? For example, is the liver sluggish, right? Is there any mitochondrial dysfunction, right? So there's no one answer. Some folks may be a lot quicker. Some folks might take longer, right? Could take up to 6 to 12 months, right? Generally, I would say you wouldn't want to retest this no later than 3 months, but that depends, right? Is there any other infections going on, right? Is there any high toxin burden load with mold or heavy metals, right? And even environmental toxins like pesticides, right? Phthalates. So until you don't clean all that up, right, you're not going to see too much improvement because you can keep throwing these supplements, but you need to stop the source. That's the most important thing, right? And that kind of leads to maybe the treatment, right? You have to look at the root causes. Oxidative stress just doesn't happen randomly. It's usually a combination of lifestyle factors, environmental, right, metabolic, genetics, right? So you really need to do a thorough assessment in those areas for the patient.
And, oxidative stress, of course, goes up with some things like, having a surgery or a urinary tract infection or having. So I'm just curious, is there a recommendation for how long you should wait after either having an infection or getting treatment before you should do a test to get a baseline value?
Yeah, so it could be both, right? You could do it before or after. It's up to the provider, right? What they're really trying to see. Yes, infections, surgical procedures, right? Tissue trauma, right? That will lead to kind of a surge of reactive oxygen species. So that can overwhelm the body's natural antioxidant systems, right? Damaging DNA, lipids, proteins, and even weaken the immune defense, right? So it just depends, right? Ideally, you might want to do it a little bit later, but you might be dealing with patients who are already immunosuppressed, right? So you just have to kind of take that with the clinical picture that if you are testing at that time, maybe the reactive oxygen markers, the biological markers might be higher, right? So there's no right or wrong way. It's just what you feel comfortable as a provider.
Okay, great. And that's actually another question is like if somebody's on anti-inflammatories, even something like IVIG, is that going to affect these markers?
Yeah, so it might, right? Definitely the inflammatory markers you want to pay attention to, right? So because, these type of drugs dampen the inflammation system, right? So that could in turn lower the reactive oxygen species, right? Now patients have to be on medications, right? They can't just take them on and off, right? Some you can. So you just have to keep that in mind when you are testing, right? If you are on some type of corticosteroids, for example, right? So you just have to kind of take a mental note with that patient that is this the true level, right? So because of the dampening of those inflammatory responses. So yes, you just have to kind of take a clinical interpretation.
Got it. So if you talk to us about like if you're a provider or clinician who's listening to this, tell us where like how they would follow up on this. And then if you are a non-clinician, if you're a potential patient or you're interested in this test, like talk to us about some future resources and where to go from here.
Yeah, definitely. Right. We welcome all providers to come join Vibrant Wellness. Definitely go to our website, vibrant-wellness.com. You can create an account with us, and then you can get access to all these different tests that you need to order for your patients. Definitely connecting with us on social media, right? You can listen to our webinars and podcasts. So all that information is on our website. Pretty much at the bottom part of the website. And once you've ordered any tests, right, we have our clinical team here to support you, right? So to go over exactly what I did, maybe a little bit more specific, right? Then we also have key resources as well. We have good interpretive guides for majority of our tests, which can be downloaded from the provider account. And that is a quick way to understand what each enzyme does, right? It's always tough to explain. There's just an exhaustive amount, how specific SNPs impact function, what risks may be present for the patient. It also guides you identifying with clinical questions, right, to ask and how to personalize treatment, right, and based on the patient's genetics. And then it's also nicely color coded, right, risk levels, so making it easier to focus on the tension where it matters most. And then within the actual report itself, the lab comments are embedded directly on each test report. So that streamlines interpretation even further. Kind of clear, easy to understand explanations, right, of what's elevated or partially elevated risk means for each SNP. And that's all written in patient-friendly language. So that's been helpful for providers.
Yeah, it's great. Yep. Good.
Perfect.
Any last thoughts about this? I mean, we really went for it.
Yeah.
Great.
Very dense topic, right? As you can
Yeah.
see, right? So this requires a lot of homework, right? And coming down to looking at the patient, right? So it's kind of a multifaceted approach, right? We talked about looking at potential toxin exposure, right? How's the metabolic health, right? Is there any insulin resistance, any blood sugar instability, right? Those can all increase oxidative stress, right? Diet, lifestyle, right? Certain foods can help raise that, right? Alcohol, right? Fried foods. So you want to focus on more polyphenol rich foods, right? Antioxidant rich foods. And then you always have to look at inflammation and infections, right? That can also drive kind of chronic inflammation goes hand in hand with oxidative damage as we discussed. So that's super important, right? And definitely genetics play a role, right? Certain genes make you more prone, as we talked about, to inflammation or oxidative stress, right? Especially if you're exposed to triggers. So lastly, right, once you've identified the likely contributors, you can build a plan for your patients to bring that oxidative stress down.
Great. 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 Thank you.
Perfect.
Thank you. Well, thank you so much for this expose today. We're really grateful for it. If you're interested to connect further with Dr. Dhanjal, then vibrant-wellness.com is just made a noise. Sorry about that. So if you're interested to connect, vibrant-wellness.com is the way to get a hold of Vibrant, and you can explore. They've got a great website with a ton of information on it. So this has been really helpful. Thank you so much.
Great to hear that out.
Thank you. Thank you. And we love that you've joined us today to discuss how to make our whole world medicine. We rise or fall together and are committed to sharing knowledge and insight that empower us and our communities towards greater health. Thank you for listening to the Neuroveda podcast for complex health, and we'll see you next time.